In Chapter 16, we're going to talk about gene expression and the regulation of gene expression, because it's not just enough to know what genes are in an organism. It really matters when and where they are expressed or not expressed. So let's look at an example of this. All cells have the same genome. In an organism, every cell has the same genome. But not all cells are the same in multicellular organisms. So let's think about a human here, right? We have what we call a stem cell. That's a cell that can become any other type of cell. So it could differentiate into a muscle cell, or a fat cell, or a bone cell, or a blood cell, or a nervous system cell, or epithelial cells, or an immune cell, or the sex cells. All of these cells are very, very different. And to get those differences, what do you think has to happen? Well, we need differences in gene expression, right? So the gene expression is the process of turning on a gene to produce an RNA and a protein in most cases, right? We've talked about genes that don't produce proteins, but we're going to talk about it in the protein coding context. So not every gene is going to be expressed in every cell type. Some genes are only ever expressed in one of these cell types. Some genes are expressed in all of them. The regulation of that, when we turn genes on and when we turn genes off, is really what makes these cells different. So this is the process of development, going from one type of cell into another type of cell. That's one reason gene expression differences need to happen. And of course, to get these changes in gene expression, we're going to need to look at our central dogma, right? Going from the DNA, transcribing that into an RNA, and then translating that into a protein. Every step where something happens, that's a potential place where regulation can occur. So let's look at an overview of regulation of gene expression. So we're going to discuss why some cells don't express all of the genes all the time. We're going to look particularly about prokaryotic gene regulation and where it occurs. And then we'll talk a little bit about eukaryotic and see that there's a lot more levels here because of the complexity that's there. So here's a question. Why regulate gene expression? I've already kind of given you one example, but think more broadly about an organism. Why might they need to regulate gene expression? Go ahead and pause the video, think about that. All right, so there's a multitude of reasons. We talked about development and differentiation in eukaryotes, but one of the main reasons that organisms regulate gene expression is to save energy. Let's think of the example of making the genes to break down lactose. Some bacteria make genes to break down the sugar lactose. If there is no lactose present in the environment, it is pretty wasteful to make all of these genes to break down lactose, right? So it would be nice if we could turn off those genes until they become necessary. We will see in great detail how organisms actually do this. Responding to the environment, right? The temperature changes, or the available nutrients change, or if you're like a pathogen, you go from outside of your host organism into your host organism and you start causing disease or something like that. There's all kinds of possibilities here for the environmental changes, right? Expressing cell-specific proteins. So if you're like a white blood cell and you need to make certain proteins on the surface of your white blood cell or whatever, or you need to make antibodies or something like that, that's your one job, and you make those proteins in that specific cell. That ties into development, as I said. And then we have this whole immune system thing, defense against pathogens, protecting yourself against harmful microbes. And in bacteria, they even have a rudimentary immune system. They make things like CRISPR-Cas9, and they also make restriction enzymes. So these kinds of things are all challenges, and genes need to be turned on and off depending on the situation. So let's think about the differences between prokaryotes and eukaryotes here. Prokaryotes, right, we said much smaller, simpler. Not that they're not complex, but they don't have a nucleus. They don't have any organelles. They can do transcription and translation at the same time. So they're all about speed, right? Whereas eukaryotes, we have many, many different cell types. We think of a plant, right? We have a lot of organelles. We have the nucleus. Transcription occurs there. Translation happens outside of that. We have chloroplasts and ribosomes and mitochondria and all these complicated organelles that are in there. So everywhere we have some differences in this system, we can potentially have points of regulation of gene expression. Let's look at prokaryotes first. The main way that prokaryotes regulate genes is through regulation of transcription. Do you make an mRNA of that gene? If yes, it generally becomes a protein. If no, you don't make that protein. So basically, to turn genes on and off, we turn transcription on and off. And I will show you some very detailed examples of that in a moment. Once a prokaryotic mRNA is made, it always basically becomes a protein. That's our pretty much safe bet there. In eukaryotes, there's a lot more levels of gene regulation here. We're going to start at the top and work our way through. This is not an exhaustive list, obviously, but we're just learning. So, really at the top, we have the signaling, right? We talked about external signals from outside the cell that come in. We're not going to get too much into that here, but there are external signals. But let's look at our central dogma here. So remember the chromatin? Chromatin is DNA that's wound around those histone proteins. And we talked about how DNA that was more loosely wound was able to be expressed, whereas if it's tightly wound, is not able to be expressed. So if you wind up the DNA, that basically turns off gene expression in that region. So parts of a chromosome can get unwound or wound. We would call this epigenetic regulation here. It's above the level of genes. And this is something we're still learning about, how this is controlled and how it changes and maybe how our actions towards our body change this. And there's even some thought that potentially basically the conditions that your ancestors lived in could alter the epigenetic state that gets passed on to you as a descendant. It's called transgenerational epigenetics. It's a little bit controversial. We don't fully understand it or have a good a grasp on it, but we're still learning. Okay, so chromatin level. Then we have control of the transcription. So can the DNA then get turned into RNA? And we talked about transcription factors being important in that. This chromatin level kind of is regulating transcription because you can't access the DNA, you can't do transcription. So you can kind of think of them together. Then we have the post-transcriptional level. This is like mRNA processing and moving the mRNA out of the nucleus. So remember we talked about our splicing out of introns and like alternative splicing. That's a form of gene regulation. Whether it becomes one protein or a different protein, that can happen there. So once it gets outside, whether it gets translated or not, so the translational level, does the mRNA get turned into a protein or not? There are not as many great classic examples of this, but we are learning more and more. For our purposes, we're not going to talk too much about translational control, but maybe one or two examples here. But we're going to focus on the transcriptional level. Then we have the post-translational level, how the protein is folded and then modified. Some proteins get made and they're in an inactive form, and like part needs to get snipped off before they can go do their job, or we need to stick something onto it to activate it. And then after the protein is there, about degrading the protein, that's basically turning it off again. So these are all levels of regulation here. So at what level does most bacterial gene regulation occur? Go and pause the video, think about that. All right, hopefully you got this one. The transcriptional level, taking that DNA, turning it into RNA. That's what most bacterial gene regulation is. All right, so all cells in an organism have the same genome, but they express their genes very differently. Gene expression, we define that as the process of turning on a gene to produce an RNA and a protein. Gene regulation happens for many different reasons. One of the primary ones is just conservation of energy, right? Not making proteins you don't need. Responding to the environment when your environment changes to be able to react to that, you change gene expression. And then in eukaryotes, we have the process of development and cell differentiation. Prokaryotic gene regulation is mostly at the transcriptional level, and we saw that eukaryotes regulate gene expression at many levels: the chromatin, transcriptional level, post-transcriptional level, translational level, and post-translational level. All right, in the next section, we're going to dive into prokaryotic gene regulation.